Hello everybody. So in this video we're going to be talking about sections 1.1 and 1.2 in the book. They cover a variety of topics. We're going to start with sampling and bias. And I may not cover the topics in the same order as in the book, but I'm going to cover all the topics that are important, just maybe in a slightly different order. Okay? So here's what we're going to talk about today in this video. We're going to talk about anecdotal evidence. We are going to talk about populations and samples. We're going to talk about bias, and we're going to talk about different sampling methods. Okay? So let's get at it. So first thing, anecdotal evidence. Anecdotal evidence is information based on accounts and narrative. Accounts and narrative, as opposed to information collected by careful and fair methods. Anecdotal evidence is dangerous because it can appear factual and therefore can easily mislead people. So we hear anecdotal evidence all the time. When somebody tells you a story about such and such happened to such so-and-so or something like that, or I heard that, you know, doing this makes it more likely for you to catch a cold, or things like this. These are usually anecdotal stories. They just come from accounts or narrative. They aren't based on scientific studies. Now I have a handout here, and I post these slides online. You can find those via the links on the course webpage. And please go through and read through that. I've got a lot of examples of anecdotal evidence there, but I'll go through some things here. All right, so here are some examples of anecdotal evidence and the faulty reasoning that could arise from them. So the first one here, we have a man on the news got mercury poisoning from eating swordfish. So the average mercury concentration in swordfish must be dangerously high. I mean, I heard this story about this guy getting mercury poisoning, so therefore we should be afraid of swordfish, right? Well, of course not, right? We don't know if if when we eat swordfish, we're also going to have the same problem this guy had. I mean, maybe he just got very unlucky. It doesn't mean that all swordfish are problematic. It might be something to look into and investigate, but this fact alone is simply anecdotal. It's just a story. So just because this man got mercury poisoning from swordfish doesn't mean you or I or the majority of the people are going to get mercury poisoning from swordfish. The only way we can, really be sure about that is if a scientific study was done and they've used careful methods to prove that you can get mercury poisoning from swordfish. So that's just an example of anecdotal evidence. We shouldn't take it too seriously. Another example. I met two students who took more than 7 years to graduate from Duke University. So it must take longer to graduate at Duke than many other colleges. Well, of course, we shouldn't draw that conclusion. Just because these two students took more than 7 years to graduate doesn't mean all students are going to take more than 7 years to graduate. But you can see, I think, how we all could fall into that belief. We might talk with our friends and they said blah, 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 and so we kind of take it to heart, right? But should we? Well, no, not until it's been scientifically studied. If someone does a sample and does under fair methods, if they prove that it takes more than 7 years to graduate from Duke, fine. But with just these two anecdotal stories, not enough information. Another one. I heard that some people who took hydroxychloroquine recovered quickly from COVID-19 infection. Therefore, hydroxychloroquine must be effective at treating COVID-19. I think you may have heard this story. A lot of people are supporting the idea of hydroxychloroquine being an effective measure to treat COVID. But no scientific study has said that it is. And so all this really is at this point is just anecdotal evidence. These are just stories. Maybe some people, like it says here in this example, did recover quickly. But do we know for sure that it was the hydroxychloroquine that actually did it? We don't. Maybe they would have recovered fast whether they took it or not. Maybe it actually made them recover slower. It may actually cause problems. Maybe they would have recovered even faster if they didn't take it. Just because we heard these stories doesn't mean that it's true. These are just anecdotal stories. These are just anecdotes or anecdotal evidence. Okay, so these are things we have to be very careful about. So how do we be more careful about this kind of stuff? Well, first we should talk about populations and samples. Okay? When we do a statistical study, we do it about the population. We are trying to draw conclusions about the population. So the population is the collection of individuals or items that a study is about. That's who we're really interested in. Now, can I go out and study the entire population? Usually you can't. Usually the population is too big, or it's too expensive, or it's just too inconvenient to go out and survey everyone in the population, or take a sample from everybody in the population, study everybody. It's just not feasible. So what do we do instead? Sample. We take a sample. A sample is a group of individuals or items from whom the data is collected. So instead of taking the data from the entire population, that's not feasible, we're going to choose a small group and take the data from them. Okay? And that might be more convenient and more feasible. And hopefully the conclusions that we draw from the sample generalize to the population. If our sample is a good one, we can say that. And we'll talk more about that later. But let's take a look at these examples here, these exercises. For each research question below, identify the population and the sample. So number 1, a survey of 847 U.S. adults found that 31% have had to put off medical care for themselves or their family in the past year due to cost. Okay, so what is the sample here? Well, let's see. The sample is the group of individuals from whom the data is collected. So who did we collect the data from? What was that percentage based on? Well, that percentage is based on the 847 U.S. adults. So that is the sample. The sample is the 847 U.S. adults. What is the population? The population is who this study is about. Well, they may have only collected the data from 847 U.S. adults, but it sounds like this study is actually about all U.S. adults. It's not supposed to just pertain to those people that participated in the survey. They're trying to draw a conclusion for everyone. We should maybe stipulate that it's everyone in the U.S., because people outside the U.S. may have very different opinions. So I would say the population is all U.S. adults. The population would be all U.S. adults. All right, let's look at another example. To gather information about starting salaries at companies listed in the Standard & Poor's 500, a researcher contacts 65 of the 500 companies. So the Standard & Poor's 500 is just a collection of 500 companies. Let's see, what is the sample here? Well, the sample is who the data was collected from. Well, the researcher only contacted 65 of the 500 companies. So the sample is the 65 companies contacted by the researcher. That's the sample, the 65 companies contacted by the researcher. What's the population? Well, let's see. To gather information about starting salaries at companies listed in the Standard & Poor's 500. So we're only talking about the Standard & Poor's 500. So that's the population. The population is the companies in the Standard & Poor's 500. Okay, next example. To determine the level of pollution in a lake, researchers analyzed vials of water filled at several different locations and depths in the lake. Okay, what's the sample that was taken? Well, let's see. Where was the data collected from? Researchers analyzed vials of water filled at several different locations and depths. Well, that's the sample. The sample is the water from the several different locations and depths. That's the sample. Not the whole lake, just those bits of water. And what were we trying to draw a conclusion about? The level of pollution in the whole lake. So the population is all the water in the lake. All right? So by studying our samples, we're trying to draw a conclusion about the whole population, which is the entire body of water, the lake. Okay? So those are populations and samples. Now, it's important when we take a sample, if the sample that we take is nothing like the population that we took it from. Maybe it's just maybe I'm sampling, I'm talking about, people's satisfaction with life, and the only people that I include in my sample are millionaires. Is that a good measure? Is that going to provide a good measure for everyone's level of satisfaction with life? Well, probably not, right? Because that sample isn't representative of everybody. Millionaires aren't like everybody else. They're special, right? And so that sample is not representative of the entire population. So it's important that we choose samples that are representative. So let's read this. A study makes a statement about the population. That's what the study is about. But the data comes from the sample. Therefore, we must try our best to ensure that our sample represents the population as closely as possible. A sample that approximates the population very closely is called a representative sample. So let's look at some examples here. So here we have a population of men and women. So I've copied the same population 3 times. But in each picture, I have a different sample. So we see that in the population, men being blue and women being red, it's about 50-50 men and women. But look at my sample, the circled dots here. My sample, is that representative of the population? Well, in the first case, definitely not. I've got more men in my sample. I have a higher proportion of men in my sample than women, whereas in the population, that's not the case. So this is not a representative sample. It's got more men than a higher percentage of men in my sample than there are in the population. The second example has the opposite problem. This sample has a higher percentage of women than men by a large margin. So it is not a representative sample, right? If I go to the third one here, this one looks a lot better, at least as far as gender goes. Okay, so I would say that as far as gender goes, the first 2 are non-representative. They're not very good. The third one looks pretty good. It's pretty representative. The population's about 50-50, and I've got about 50-50 in my sample. That's a representative sample as far as gender goes. So that's what I'm saying down here. As far as gender is concerned, the sample on the right is the only one that's representative. However, further scrutiny may be necessary if other factors are relevant, such as age, race, income, etc. And if you're doing a survey about, like I said, satisfaction of life, those things, income, and in particular, might be something that you want to make sure you've captured in a representative sense in your sample. So the primary goal in collecting a sample for a study is to ensure that the sample is representative. If it's not representative of the population, then why should we be able to draw any conclusions from it? You're just talking about something else. All right. One other issue that we can encounter when we do studies is bias. Now, it's very important not to confuse bias with error, and it's very common to confuse these. So bias is the systematic tendency to produce a particular type of error. Okay? Errors happen all the time. Error is unavoidable in pretty much any study, but errors that are not prone to repetition tend to get washed away in the average. Right? So if I make an error here or there, a little bit wrong here, a little bit wrong there, if I have a large enough sample, those errors probably aren't going to add up to much. They kind of even out, right? But the more data we collect, the less impact such errors have. Now, bias doesn't work that way because bias is systematic. So it's going to cause that one type of error to occur over and over and over again. So no matter how much data I collect, it's always going to be there. It's not going to get washed out. All right, so bias is something we have to be very careful about. Errors are going to happen, but they aren't necessarily bias. Bias is when the errors happen systematically. And the best way to deal with bias is to prevent it from occurring in the first place. And the best way we can do that is by identifying and thinking about what kinds of bias there are and just being aware of them. Okay? So here's some types of bias. Now, if you look in the literature, there's lots of different kinds of bias. Oops. I've seen lists of like 20 or more classifications of bias. I'm just listing 4 here. So let's just look at these 4, which I thought were particularly important. Sampling or selection bias. This is the tendency of a sampling procedure, the way we collect our sample, to include or exclude certain types of individuals. Okay, so for instance, if I was doing a study on, again, satisfaction of life in New York City, maybe I'm going to take a bus up the Upper East Side and survey people as I go. Right? Is this going to be, is this going to have any errors in how I get representative people from the population of New York? Well, I think it would. If I just go up on a bus on the Upper East Side, I'm probably excluding quite a few groups that live in New York City from my sample. So there's a systematic error. I'm systematically excluding certain types of people in that sample. And so this is sampling bias. Maybe it was accidental. Maybe I'm just being a jerk in the way that I'm conducting my study, but I've chosen a route that is systematically excluding certain types of people from being in my sample. That's bias. Another type of bias is confirmation bias. And this is something that I think we all do. You have to admit, everybody does this. It's a tendency to search for, interpret, favor, and recall information in a way that confirms one's beliefs or hypotheses while giving disproportionately less attention to the information that contradicts it. So in other words, we hear what we want to hear, right? So, if somebody is a supporter of Trump, they will tend to focus and hear the news about how he's doing a good job. And if people are against Trump, they will probably focus on the news that indicates that he's doing a crummy job, right? And both sides probably tend to ignore the things that contradict those beliefs, right? So, and confirmation bias, you have to just admit that it happens to everybody. We always have to be a little bit careful about it. It's so easy to just kind of ignore what you don't want to hear, right? So you always have to be very careful to try to fight that bias and put measures into your statistical study to kind of make it hard for that to happen. Observer expectancy effect. This occurs when researchers' expectations cause them to subconsciously influence the people participating in an experiment. So this might happen in a hospital if a doctor is giving out they might be giving out medicine to different people, and he might be giving medicine that works to some people and medicine that doesn't work to other people. The medicine that doesn't work, they do this. It's called a placebo, and it's so that we can measure the difference between the two groups, and we know that it wasn't just the people reacting to the fact that they took a medicine that made them get better. We know that it's actually because the medicine was effective, not just the fact that they took it. So we have to give half the people some fake medicine. Sounds unfortunate if you happen to be in one of those groups, but it's a way of fighting this other kind of bias that those people might undergo, where they might believe they should get better because they took a pill. You need to control for that. But the problem is that the doctor may treat the patients he gives the real medicine to slightly differently. He might be more hopeful for them. And so by the way that he behaves to those patients, his behavior may carry over and cause an effect in their recovery, right? And that would be an example of this. And then reporting bias. And reporting bias is the selective revealing or suppression of undesirable information by subjects or researchers. So in other words, we exclude the stuff that we don't like, that isn't good for proving what we want to prove. This doesn't help prove my research? Let's just ignore it. Now that sounds pretty sinister, but sometimes we do this by accident, right? You might be, a researcher may have to take a sample of like a tissue sample of like a piece of a liver or something, and then they have to count the cells that look problematic. And now there's the judgment call there. Which cells look problematic, which ones look like they're bigger than they're supposed to be, or something like that. There might be some borderline cases. Now, if the research is trying to prove that there's more problematic cells than usual, on those borderline cases, that researcher might say, I'm going to count that as a problematic cell, because it helps their cause. It helps them publish their result. and that would be reporting bias. So they'd want to devise a way to count the cells that wouldn't allow that to happen, right? To fight that reporting bias. And finally, the last bit here, sampling methods. Now, the reason we have sampling methods is to fight bias, fight sampling bias in particular. We want to collect our sample in a way that makes sure that it's not going to be biased. Okay, so one common method that we'll be talking about a lot in this class is simple random sampling. A sample is referred to as simple random sample if each case in the population, each individual, has an equal chance of being included in the final sample. So everybody's got an equal opportunity to be included in the sample. We're not excluding anybody. And we're not making it more likely for some than for others. And knowing that a case is included in a sample does not provide useful information about which other cases are included. So just because I'm in the sample doesn't mean that my friends are going to be in the sample, right? This is called simple random sampling. Everybody has an equal opportunity to be in the sample, okay? So one very straightforward way to do this is to just assign each member of the population a number. It doesn't matter how you assign the numbers. You can do that systematically. You can assign the numbers however you want. You can assign your best friends your favorite numbers and the people you hate your least favorite numbers. That part doesn't matter, because what you're going to do next is you're going to have a computer select random numbers, and computers are not biased. So no matter how you set up those numbers to begin with, because the computer is going to create a random selection, it's going to produce a simple random sample. Everybody will have an equal chance of being in your sample. And you can see the little diagram here. We had our population, we numbered them from 1 to 12, and the computer picked numbers 2, 5, 8, and 10. All right, so we have a simple random sample. Now, is it guaranteed that every simple random sample is going to be representative? No. You could just by chance end up with all red individuals here in the sample. But it's not, that's a sampling error, but it's not a sampling bias, because that systematically will not happen, right? Simple random sampling is an effective way to avoid sampling bias. Now, in the book, they talk about other types of sampling, and these are really important if you get into taking samples. I'm not going to dwell on these. I'm not going to test you on these. But there are other types of sampling methods that kind of ensure that kind of thing that I was just talking about. Maybe we wanted to make sure that the sample had an even distribution of red, green, and blue here, right? There are ways to do this. And these different ways, that one would probably be stratified. There's also cluster sampling and systematic sampling. These are different sampling methods. I'm not going to really dwell on these. So that is the information for this topic, and I will see you in the next video.